Okay, so today we're talking about genetic mutations, and so I'm just going to go a brief overview. I'm not going to go too in depth about it. But basically, there are three types of genetic mutations: substitution, insertion, and deletion, and they kind of do what they say they do. In a substitution, just a base sequence. So if I have a sequence A, and then it goes mutated into ATA or something like that, that would be a substitution. The thing was just switched. Now, if I have an insertion, would be like A, and then I go to AGAA, or AGAA, and then deletion would be going to AA, and it got rid of one. And so these two are a bit more dangerous because it causes a frame shift mutation. And so in class when we learned it, we had this chart, but basically what a frame shift does is when you have your, you read three bases, you read three bases, you read three bases, when you have your start promoter region over here. But if you delete one, then the rest of the reading frame, so then it goes 3, and then technically there's 2 that you should be reading, but really it's going to read 3, so then the rest is off from, and so the reading frame of the codon's shifted one in either. If you insert, it's going to insert one and delete it. And when you do that, all your mRNA is then going to get messed up. And so here, like look, all of them changed. And so that's when the change is more drastic, you have a higher chance of it not working. And so the mutations, they do cause a variety and possibly adaptations and things to arise in the gene code, a genetic pool to allow an animal to have more fitness and to survive better. But really it's only going to be a substitution. Maybe if you switch one amino acid out, then it's okay. And then substitutions, some can be better, some can be worse. So if I have an AAA sequence, I'm going to draw 3 of AAA, and we remember referring back to this chart here. If I have AAA is going to code for AAA, lysine, LYS. But if I turn that to AAG, oh cool, it still codes for lysine. This to this, that's fine. Now, if it goes from A to AA, look, if we look to C or U, if we go AAC, then it changes to ASN. So here we have a change in an amino acid, but only one amino acid's been changed, the rest will remain the same. If we change the second letter, however, we go AGA, what does that get us? AGA. So now, once again, we are on ARG. So once again, we change the amino acid, but only one's been changed. And then if you change the first one, so instead of AAA, we go UAA, UAA. Oh, that's a stop. So this kind of would mess it up a lot, because you may have the entire rest of the thing, but the ribosome's just going to stop putting them in and be done making the protein chain, because that's what it was told to do. And so on a substitution, you can really have different varying results as far as how bad it is for you. This could be really bad, and these could be less bad. And then these ones are usually decently bad because you're shifting the entire reading frame for that codon set. So I hope that was helpful.